Let's start again. So discontinuous variation is caused by a single gene. Continuous variation is caused by polygenes. And there are a number of ways you can have genetic variation. The first one is by meiosis. Meiosis happens in prophase 1. The genetic variation happens in prophase 1 and metaphase 1 and 2 of meiosis. Apart from that, you'll also have genetic variation because of fertilization, because remember that there are many sperms that are made by humans and many eggs. So a different egg can mate with a different sperm and fertilize. This will cause genetic variation. And apart from that, you'll also have genetic variation due to mutations. Mutations are the result of new alleles. And also remember that phenotype is the result of interaction of genotype and environment. So phenotype depends on the genotype and the environment, and the factors that will change the phenotype is number one, nutrition. So the diet that you eat, if you eat more, you'll be more healthy. That means you'll look more fat. And apart from that, there is water availability. So if you drink water, if you have the amount of water in your body will also affect the phenotype. The disease that you have, if you have a disease, that will also affect the phenotype. And apart from that, the environment that you're living in. So the temperature, the light, chemicals, all of that will also affect phenotype. And remember that environment effects usually are greater on polygenes. So if a phenotype is affected by polygenes, meaning multiple genes, then the effect of environment on these polygenes would be greater. And remember that polygenes are many genes controlling one trait. So for example, 3 genes controlling for the skin color. So if your environment affects your genes, these 3 genes, then the effect of this environment would be greater on your skin. And yeah, so phenotype is affected by the environment, and remember, phenotype will show continuous variation. So let's move on to definitions. What is a population? A population is a group of individuals of some species living in the same area that can interbreed. And alleles are different form of a gene. And allele frequency is the number of occurrences of allele in a population. So the number of similar alleles that you have in a population is called allele frequency. And a gene pool is all the genes present in a population. So all of the different kinds of genes you have in a population is called gene pool. And overpopulation is when organisms produce many offsprings due to great reproductive potential. But the population size will remain constant over time. So over time, this overpopulation would decrease. So over time, you'll have the population size will become constant. Why is this? This is because of competition for survival. And remember that selective pressure will always act on populations. Selective pressure is the environmental factors that cause different survival between individuals with different traits. So a selective pressure, if a population has selective pressure, then remember, this will cause the population to be reduced. And also, variation is caused by a change in phenotype. So if you have a change in phenotype, this will cause variation. And remember, variation will cause competition. So there is a theory called survival of the fittest, which says that some species have advantageous alleles. For example, some people are more light-skinned, and this will have a different effect on the sunlight that you absorb. So if you are more dark skinned, this means that you'll absorb more sunlight. So this is just an example, but what this causes is some people are more adapted to survive than others. If you absorb more sunlight, right? If you absorb more sunlight or UV light, you have a lot more chances of developing skin cancer than if you're light skinned. So this is an example of survival of the fittest. So higher fitness means you're more likely to survive. And if you're more likely to survive, this means that you reproduce more. And this also means that you'll pass on the advantageous alleles to the next generation. So if you are dark skinned, if you are light skinned, then you're more likely to survive the UV light. And this means that you'll more likely to reproduce and pass on your genes. Which genes? The genes that code for your skin color. So yeah, this is called survival of the fittest. And over many generations, this can cause a change in allele frequency. And this means that when you have a population, over time, right? Over time, this population can change its alleles frequency. This means that over time, the frequency of light skin color will increase in a place where there is a lot of sunlight. So frequency of advantageous genes will always increase over time. This will result in something called adaptation. And adaptation is a structural behavior or psychological trait that is a result of natural selection over time in a species of a population. So remember, all of this survival of the fittest is caused by natural selection. All right. So yeah. And this can lead to something called speciation, where speciation is a formation of new species. We'll talk about more. We'll talk more about speciation, but yeah, just know that speciation is the formation of new species. And so there are many types of natural selection. Remember that selective pressure will cause natural selection, and favorable alleles will be selected for, but less favorable alleles will be selected against. What does this mean? I told you earlier that in a place where you have a lot of UV light, the light-skinned people will more likely to survive. And so the the alleles, the favorable alleles are the alleles for the light skin, but the alleles that are not selected uh, are the skin color which will affect more on UV light. So there are three types of natural selections that you need to know. Stability selection is the first type of selection. And in stability selection, remember the intermediate is selected and the two extremes are selected against. So the intermediate basically means that the gene is better in both ways. So the shape will look something like this. The shape will look something like this. This is stability selection. The second type of natural selection is directional selection. Directional selection is when one extreme, so one extreme and one intermediate is selected. Ignore this. One intermediate and one extreme, and the shape would move towards the right. So the shape will move towards the right. The curve moves towards the right. So one extreme and one intermediate is selected, but the other extreme is selected against. And the third type of natural selection is disruptive selection. In this type of selection, in this type of selection, you'll have the two extremes that are selected and the intermediate is not selected. So in disruptive, you'll have the two extremes selected, but the intermediate would not be selected. So the curve looks something like this. All right. And yeah, so moving on with genetic drift. Genetic drift is when there is a random process due to chance, unlike natural selection. And in genetic drift, you'll have a change in allele frequency, the effects. And remember that there are two types of genetic drift. The migration, which is called founder's effect, and the second type of genetic drift is called the bottleneck effect. And this type of effect is caused by natural disaster. So what is the founder effect? The founder effect is caused due to migration. So few people will move to a new region, and these new people will be geographically isolated from the larger population. So for example, if you have two islands, and from one island, a few people move to this new island, and this island would be now this new island would have less population, and this island will have more population. More number of people would mean that you'll have uh, a greater genetic variation. So remember this, that uh, the founder effect will cause genetic variation, and it will also cause it's also a cause for genetic drift. Genetic drift basically means that you're changing the allele frequency, because these people can carry many different genes, and when these many different genes move to a newer population, then you'll have genetic drift. This means that this population will have a different number of genes, because new genes were introduced to this population. So yeah, this is called the founder effect. And the bottleneck effect is when you have a large decrease in genetic diversity. So this will be caused due to large decrease in population number. And this can only happen when you have overhunting, or you can have natural disaster like earthquakes or tsunamis and all of that stuff. So yeah, bottleneck effect causes decreases the population, and it can result due to overhunting or natural disasters. So in bottleneck effect, you'll have a small group of survivors, and gene pool will not be represented by the original population, because remember that the original population decreased a lot. So this will cause a decrease in gene pool. So, all right. So yeah, remember these two effects, bottleneck and the founder effect. Now let's move on to artificial and natural selection. Artificial selection is caused by humans, and the selective pressure are humans. Whereas natural selection is when is caused by the environment or the nature. And the selective pressure here in natural selection is environment. And artificial selection is for humans benefit. So if a human wants for example, if a human wants more crops from a crop field, he'll use artificial selection, and this means that it's for his own benefit. But natural selection is for the organism's benefit. So if the organism wants to survive, then natural selection will happen. And artificial selection causes inbreeding, but natural selection causes outbreeding. Inbreeding is when the species from the same population, they breed together. And why is inbreeding bad? Because inbreeding can result in uh, the recessive alleles being described, uh, being shown. So inbreeding is bad because it results in recessive alleles being expressed. So artificial selection can cause inbreeding, because remember that uh, if you want better crops, then you'll cross the crops that are better. So humans won't look at if the species or the population are the same or not. They'll just inbreed. But natural selection causes outbreeding. And outbreeding is good because it causes the dominant allele to be expressed. And also remember that artificial selection causes a decrease in genetic diversity, but natural selection causes uh, the genetic diversity to remain high. And also, artificial selection increases homozygosity but decreases heterozygosity. Zygosity is when the species have the same alleles, and these alleles can be recessive as well. This is why artificial selection is bad. So yeah, but natural selection causes increase in heterozygosity and decreases homozygosity. And yeah, so let's talk about artificial selection first. It is caused by human, as I said, and the selective pressure in artificial selection are humans. And so yeah, humans choose parents with desirable features and crossbreed them, select offsprings with desirable features, and this they'll reproduce these features for many generations. This increases allele frequency for the desired trait, but decreases the allele frequency for the undesirable trait. So, artificial selection, remember, it will cause an increase in the desirable trait. So, yeah. And there are disadvantages to this because, as I said, it will cause inbreeding. It will cause increase in homozygosity. Inbreeding causes harmful recessive alleles to be expressed, and this will cause inbreeding depression. And this also, artificial selection also causes limited gene pool. Limited gene pool basically means that the number of genes in a species is reduced. This means that they are not well adapted to the environment. So yeah, and so let's move on to evolution now. We'll talk about natural selection later on. Evolution is the idea that organisms change over time. So species have evolved over time from pre-existing species. Organisms are descended from a common ancestor. Evolution means that organisms are descended from a common ancestor, and the more closely related the species is, the more recent the common ancestor is. And we can figure evolution out. We have proof of evolution by two evidence. The first one is the amino acid sequence of the protein. We can figure out if the ancestor is common to us or not by looking at the amino acid sequence of the two different species that we are comparing. So the more similar the amino acid sequence is, the more closely related the ancestor is. So you can see that uh, mouse and rat are very similar. They have a very similar amino acid sequence. So they share a more common ancestor. And the second type of evidence we have for evolution is through mitochondrial DNA or mtDNA. mtDNA. So we compared the nucleotide sequence in the mtDNA of two different species that we want to compare, and the less difference there is in the nucleotide sequence, the more closely related the species is. Uh, why is the mitochondrial DNA good for comparing species? This is because mitochondrial DNA is circular. This means that it doesn't undergo crossing over. Uh, the mutations in mtDNA are constant, and mtDNA mutates faster than the normal nuclear DNA, and changes only arise by mutation and are not repaired. So a good uh, a good way we can use mtDNA for evidence is because mtDNA is, remember, it's not associated with histone protein. Because if there is histone protein, then the DNA can be repaired. But empty DNA is not repaired because it doesn't have histone protein. And this is good because this means that there will be less changes in the DNA structure. So we can figure out if they share a common ancestor or not. And another reason it's good, empty DNA is good, is because it's smaller. Remember, empty DNA is smaller than normal DNA. This means that it has fewer genes. And if you have fewer genes, you can analyze them much quicker. And the last thing, empty DNA is passed from mother to offspring. So all descendants of the females will have identical empty DNA. So yeah, and let's move on to species now. Species are a group of similar organisms which have the same morphological, psychological, behavioral, and biochemical features. So remember these four features of a species, and they can interbreed to produce fertile offspring, and they are not reproductively isolated from each other. And so speciation is the formation of new species. Speciation involves a reproductive barrier between population, because the populations cannot interbreed. This causes speciation. And remember that uh, a good reason for no interbreeding is caused by reproductive barrier. So there are two types of barriers: the pre-zygotic isolating mechanism and the post-zygotic isolating mechanism. And the pre-zygotic, from its name, happens before the fertilization, and post-zygotic happens after the gametes are fertilized. And in post-zygotic, you'll have uh, the the species cannot reproduce. So this is post-zygotic. And there are two types of speciations that you need to know. The allopatric speciation uh, is caused by geographical isolation. So when two species are geographically isolated, meaning, let's say this is one species, this is another, and they are isolated by a big river, then there will be speciation. And which type? Allopatric speciation. But you'll have sympatric speciation in the same location. So sympatric happens at the same location. Allopatric happens at two different locations. So let's talk about the reproductive barriers that the species have. The first one is pre-zygotic isolation. Remember, this happens before fertilization. It can be due to a number of reasons. The first one is caused by geographical. So populations are separated by geographic barriers. So it can be mountains, rivers, or any type of geographical isolation. And if geographically isolated, then you will have pre-zygotic isolation. And the second type is temporal. This means that populations breed during different seasons of the year. This will also cause pre-zygotic isolation. An example of this is birds. Some birds like to breed in winter, but some like to breed in the summers. So this can cause pre-zygotic isolation. And behavioral isolation is also pre-zygotic, meaning populations have different mating preferences. This means the example of this is when a species is more attractive or less attractive. So attractiveness can affect behavioral pre-zygotic isolation. And the last type of pre-zygotic isolation is morphological or psychological. This is when the reproductive features do not make it possible for people to breed together. So yeah, you have to know these four types of pre-zygotic mechanisms. The post-zygotic isolating mechanism prevents the development of embryo. Uh, this can happen due to gamete mortality. So at times, gametes don't survive until fertilization happens. So the survival rate of sperms in humans, if it's low, then you cannot have fertilization. The second type is zygote mortality. Sometimes the zygote is formed, but cell division doesn't start in zygote. This is an example of post zygotic mechanism. And the third type is when the adult offspring is unable to produce gametes, and this can only happen in humans, mammals, and plants. So when humans and mammals, they don't have reproductive mechanisms, meaning then they cannot produce gametes, and therefore they cannot fertilize. So yeah, this is post-zygotic and pre-zygotic isolating mechanisms. So let's move on to the different type of speciations. The first one is allopatric. So remember, allopatric speciation happens due to geographical barriers between the same species. So geographical barriers can be anything. So rivers, mountains. When two populations are physically separated, this means that the two populations cannot breed together. So an example of this would be one organism from here cannot breed or cannot breed with the organism from this location. So no breeding happens between the two populations. This means that there will be no gene flow. And this also means that they are reproductively isolated. And allopatric speciation, remember, is caused by pre-zygotic gotic isolating mechanism. So pre-zygotic is caused by allopatric speciation. And there will be different selective pressures acting on the separated populations. If these two populations are at a different place, they will have different types of environment. Maybe this would be more hot compared to this, or you'll have more sunlight. The point is they'll have different environmental conditions. So this causes beneficial alleles. Individuals with beneficial alleles will be selected for, and this means that they will be more likely to survive and reproduce more. And they'll also pass on these beneficial alleles to the offspring. So over time, over many generations, the organisms from this species will be adapted to the environment, and they'll have alleles that are more benefiting to this location. So they'll pass on these alleles to their offsprings. And this causes a change in allele frequency. Remember, natural selection will cause a change in allele frequency. And because you have different environment, there will be different mutations happening between the alleles. So at these two different locations, they'll have different mutations as well, because mutations can be affected by environment. And what do I mean by affected by environment? If you have UV light, if you have more sunlight here, then you'll have more mutations happening. So yeah, this is an example. And this causes genetic drift, because genetic drift is when you're isolated from one population. So genetic drift happens due to mutations, and you'll have separated populations. And over a long time, populations have many differences, so they are unable to interbreed or reproduce together. And when they are reproductively isolated, when these two species are reproductively isolated, this will cause speciation. So the moment that species have different reproductive mechanisms, they'll have speciation. So yeah, this is allopatric speciation. Sympatric speciation happens at the same location. There is no physical isolation, so no geographical barrier. And the different features of or behaviors within a population causes interbreeding to stop. So if you have different features in two species, they'll stop interbreeding together. This causes gene flow to stop. So genes will not flow between species. And over time, there is a change in allele frequency, and this will eventually cause genetic drift. Genetic drift is when you have a big change in gene pool. And this causes population to become reproductively isolated, and eventually this will lead to a different species. The main point is sympatric speciation happens at the same location, and allopatric happens at different locations. And yeah, with this we finished all of this chapter. We'll do mutations in the next chapter. All right. So yeah, see you in the next class. Bye.